Our next presenter is Courtney Wright from Biomedical Sciences.
Good afternoon. I'm a neuroscience PhD student, and what our lab is really interested in doing is understanding what contributes to psychiatric illness so that we can better treat these disorders. What I started my thesis research with was actually the very beginning of the lifespan, so the time that you all spent in the womb. This is because during this time, insults like inflammation or poor sleep that happen to pregnant people actually significantly increase the risk of their offspring to develop psychiatric illness later in life. But we don't know exactly how this happens, and so we can't really mitigate this risk. Our lab then really tries to think about what overlapping mechanisms might exist between inflammation, poor sleep, and mental health, and we've become really interested in a neuroactive metabolite called Kynurenic acid. This is because we actually know these prenatal insults directly activate the kynurenine pathway. This is the main metabolic pathway of the amino acid tryptophan, and this elevates kinin levels in the brain. And patients with schizophrenia and bipolar disorder, severe psychiatric illnesses, have elevated brain kinin levels. And kinin not only is really important for neurodevelopment, but it actually prevents neurotransmission that's also important for learning, memory, and sleep, which are symptoms that these patients experience that are not resolved by currently available antipsychotic medications. So we really think that this elevated brain kinin contributes not only to the development of these diseases, but the lived symptoms that these patients still suffer with. So for my research, I really had to start at the basics and develop a prenatal model or preclinical model that would allow us to mechanistically investigate these questions. So in pregnant rats, I fragmented maternal sleep using automated sleep restriction chambers, and this caused major sleep disturbances at the very end of pregnancy. And in these moms, we actually saw inflammation in the blood, which can activate the kynurenine pathway, but we only saw elevated kinin levels in the fetal brain, which indicated to us that the babies at this time are actually particularly vulnerable. And so to investigate that elevation of kinin a little bit more closely, we utilized a second model where we supplement the maternal diet with kynurenine. This is because kynurenine will cross the placental and the blood-brain barriers and be elevating kinin levels directly in the tissue. And in this model, we don't see any peripheral inflammation, but we do have elevated kinin levels, and we do still see minor sleep disturbances. So what we're really interested in doing is really leveraging this foundational information to not only understand the consequences to male and female offspring across the lifespan, but really leverage the unique advantage of preclinical models to test novel therapeutic strategies, perhaps targeting elevations in kinin levels that can really promote long-term offspring psychiatric health. So thank you.